Hello everyone, my name is Maryane Oliveira. In this presentation, I will discuss one of our research conducted at the University of Illinois, investigating the optimal tryptophan to lysine ratio for 25 to 40 kilograms body weight growing pigs fed diets containing 35% distillers dry grains with solubles. I will start by giving a brief background. Distillers dry grains with solubles is a byproduct from corn resulted from ethanol production. Because there is a fermentation in the process, DDGS has higher concentration of crude protein and amino acids than in corn, which turns this byproduct to be good and inexpensive feed alternative to include it in pig diets. However, if you take a look at this graph, the concentration of tryptophan is low in corn and corn byproducts, whereas lysine is high in corn and becomes higher in the respective byproduct. For this reason, if DDGS is included in growing pig diets, the tryptophan becomes the second limiting amino acid after lysine. As lysine, tryptophan has also an essential role in protein synthesis as well as immune responses. In addition to protein synthesis and immune system, tryptophan also works in feed intake regulation, being the precursor of serotonin. As tryptophan shares the same brain transporters as lysine, excess lysine may reduce tryptophan transport to the brain for serotonin synthesis and affect feed intake. Thus, tryptophan requirement may be increased if large amounts of DDGS are used. Therefore, one experiment was conducted to test the hypothesis that tryptophan to lysine ratio may increase for pigs 25 to 40 kilograms body weight fed diets with excess lysine from distillers dry grains with solubles. Moving on to the experimental diet, a basal diet was formulated based on corn DDGS, soybean hulls, and soybean meal, which contained 15% standardized ileal digestibility of tryptophan to lysine ratio. 4 additional diets were formulated by adding crystalline tryptophan to the basal diet, in order to have 18, 20, 23, and 24% SID of tryptophan to lysine ratio. Thus, there was a total of 5 diets. Now, looking at this graph, yellow line represents the percentage of amino acids that met the NRC requirement. For tryptophan, which is in blue bars, the first diet was below of the requirement, whereas the subsequent diets were over of the NRC requirement. However, lysine concentration was about 150% of requirement in all 5 diets. Talking about experimental design, a total of 120 growing pigs with an average of initial body weight of 26 kilograms were allotted in a randomized complete block design with 5 diets and 8 pens per diet. There was 3 pigs in each pen. The experimental period was 21 days, and blood samples were collected at the end of the experiment to analyze for plasma urea nitrogen. Initial and final body weight were recorded on day 1 and day 21, and the amount of feed consumption was recorded through the experimental period. Average daily feed intake, average daily gain, and gain to feed ratio was calculated as well. For a statistical model, Data were first analyzed using orthogonal contrast to see the linear and quadratic effects of tryptophan in the diets. Then, the average daily gain and gain-to-feed ratio was submitted to linear broken line and a quadratic broken line regressions analysis in order to calculate the optimum tryptophan to lysine ratio. Now, let's move on to the results. First of all, I want to point out that for all graphs, the y-axis represents the response parameter and x-axis represents the level of SID tryptophan to lysine ratio. The average daily feed intake increased linearly as the SID tryptophan to lysine ratio increased, up 23 tryptophan to lysine ratio, and had a plateau after this level. The same was observed for average daily gain, which increased linearly as the level of SID tryptophan to lysine ratio increased, up 23%, but had a plateau after this level. Likewise, an increased gain-to-feed ratio was observed as tryptophan to lysine ratio increased, up 20%, and had a plateau after this point. Now, looking at final body weight, increasing levels of tryptophan increased the final body weight as well. But after 23% of tryptophan in the diet, there was also a plateau. These results indicated that indeed tryptophan concentration in the diet affect growth performance of growing pigs if they are fed with high concentration of lysine. Now, talking about plasma urea nitrogen, there was a reduction in plasma urea nitrogen as the level of tryptophan increased in the diet, indicating that protein synthesis has increased in the pigs. Now, let's take a look at the estimation of optimal tryptophan to lysine ratio. The blue lines represents the linear broken line and the orange line represents the quadratic broken line. Using the average daily gain as a response parameter, the estimation by the linear broken line was 20.9%, whereas the estimation by the quadratic broken line was 23.4%. Using the gain-to-feed ratio as a response parameter, the optimal tryptophan to lysine ratio estimated by linear broken line was 18.7%, whereas the estimation by quadratic broken line was 20.2%. Results from this experiment indicated that the optimal SID tryptophan to lysine ratio was 20.8%. This value is a result of the four values found in the regression analysis. Diets with high lysine may reduce the feed intake by pigs if dietary tryptophan is not supplemented, and diets formulated with 35% of DDGS for growing pigs diets may need more dietary tryptophan. With this presentation, I want to make acknowledgement for Yvonne for the inputs and financial support, and also to the whole team of Stein Monogastric Nutrition Laboratory. Thank you for your interest in this presentation. And if you would like to know more about other research topics in nutrition, you can always visit our website at nutrition.nsi.illinois.edu.